All right, please sit down. Let's start. Okay, we can start. Okay. Now, next speaker is Peter Baas.
Okay, I thought what I would do is just start out with a mention of something I think that's really important to all of us as biomedical researchers. And that's the link between basic science and the disease and injury that we heard. When I started in biomedical research, it was 20, 25 years ago. My commitment at that time, like many of the investigators you've heard from, was really in the arena of basic science. But my funding from the beginning has been primarily from NIH. Now, NIH in the United States is the National Institutes of Health. And if you look at the mission of the NIH, the mission is the healthcare, the development of treatments and cures, and elucidating the underlying etiology of diseases that affect the human population. So that being the case, why is it, do you think, that over these past decades, NIH has given millions upon millions of taxpayer dollars toward understanding things like centrosome duplication and issues that may seem very separate from human health? Well, I recall in the old days of decades past, when I would write an NIH grant, I'd have to insert a few sentences. I'd dig into the literature and try to find reference to some microtubuleal abnormalities that I could make a mild sort of argument that understanding microtubules and neurons would somehow help people with certain afflictions. But really, the genius of the approach has come to fruition. And really, the genius was that before we could really understand human disease, before we could really understand how to treat it, or cure it, hopefully someday, we're going to have to understand those underlying mechanisms. How do dendrites grow? How do axons pathfind? How does axonal transport work at the mechanistic level that we've heard from these past few days? Once we understand, and without understanding those underlying mechanisms, then we're at severe disability to really craft, to really brainstorm the kinds of therapies that would truly help patients. We could take the approach of the ancients and go through thousands of potential therapies of different sorts of extracts from plants and try to, by the process of elimination, figure out what might possibly help patients. But alternatively, if we can elucidate those mechanisms, then we can really craft strategies. So even for those of us who came into this as basic scientists. I think this basic science is really a crucial linchpin in ultimately solving the mystery of these diseases. I moved about 12 years ago. I started out at the University of Wisconsin, and I was there for 10 years. Pretty much all of those 10 years, I was a basic scientist. And about the time that I moved to Drexel 12 years ago, I think at that time, at least personally, I could begin to detect change going on. I began to submit to some various foundations in addition to NIH. Because at some level, you have to, in order to keep your lab going, you have to seek financial support from various agencies. So there were agencies such as the Alzheimer's Association, and there was the Christopher Reeve Association, which supports spinal cord research. And I began to think creatively about how the body and my research over the past decade could possibly help people, say, with spinal cord injury or Alzheimer's disease or other sorts of degenerative diseases. I also began to appreciate, looking into the literature, how now, after decades of work in laboratories all across the world, how now we can understand at a level we never could before how some of these diseases actually work, and really how many of these diseases, particularly in the case of the nervous system, really have their heart, underlying mechanism, with these microtubules that I've been studying all of these years. Lissencephaly, for example, is a birth defect that occurs with failure of the brain to appropriately laminate, to appropriately layers of the cortex. Lissencephaly is caused by mutations to LIS1, which is a protein that regulates microtubules. As we've heard about in Alzheimer's disease and other so-called tauopathies, the microtubule-associated protein tau goes around, and this can cause problems. I'm going to talk in just a very few minutes about a disease called hereditary spastic paraplegia. The chief cause of hereditary spastic paraplegia is mutations to spastin, and that had been known for a long time, before it was discovered that spastin is a microtubule-associated protein. So what I'm going to be talking about during the next hour or so is microtubule-associated proteins. And in the previous talks that I've given to the school and the workshop, I've introduced the idea of microtubule-associated proteins. So that will now be the focus of today's presentation. But also part and parcel of the presentation today, what I hope to drive home is a mixture of basic science and then disease and injury, to see that these two arenas of biomedical research are really not at all separate, but are intimately linked to one another. So let me start out by just reminding you of these incredible cells, neurons, that are arguably the cell type whose polarity, whose exaggerated morphology, across nature are the cell type whose morphology, whose asymmetry is most intimately related to the functions that they serve. Now, we all are partial to the cell type that we study, but I think I'm justified in arguing that in terms of cytoskeletal challenges, neurons are the most interesting, the most challenged. We really have to come up with the most creative solutions in order to elaborate microtubular arrays, other cytoskeletal arrays that are sufficient to meet these challenges. And in particular, neurons aren't just polar, they're bipolar. They generate multiple types of elongated processes. They have the axon, multiple dendrites. And these processes are different from one another, all the ways we've been discussing these past few days. Axons being long, slender processes, highly specialized to transmit information over long distances. And dendrites being multiple in number, thick, tapered, highly specialized to receive and process information. And so many of these challenges, the architectural challenges, transport challenges, are fulfilled by microtubules. This is another schematic that illustrates some of the things axons have to do. A couple of things I want to stress is that axons grow during development, but they also retract. Now, retraction is not just a pathological event. Retraction is also a highly physiological event. I'll talk more about retraction at the lunch when we talk about proteins. But when the nervous system develops, the number of axons, axonal branches, really overshoots. Nobody quite knows why. There's more axons than the developing nervous system needs. And a great deal of pruning back of retraction has to occur in order to ultimately result in the fully wired, appropriately wired adult nervous system. And that structure at the tip, the growth cone that we've talked about a bit, that's a highly motile structure that forages through the endothelium. It knows where it's going. It has turns to take. The movement of the growth cone is an actin-based event, but the turning of the growth cone, that depends on microtubules. So axon growth is microtubule. Axon retraction we'll talk about the role of microtubules. And then growth cone turning. A critical part of the talk for this morning is axonal branching. So another thing that happens is that developing axons will form branches. There's really two types of branching. One is bifurcation of the growth cone, just simply splits, or multiple branches emerge from the tip of the growing axon. But the type of branching, I think, that's more predominant in the case of many sorts of neurons, and I think more interesting, is called either interstitial branching or collateral branching. And that sort of branching occurs along the side of the developing axon. We'll be talking this morning about the mechanisms of that branching. So, with the process of microtubules separating, there are some basic science issues I'll be talking about. Branching is one of them. But also the disease situation. As I mentioned, hereditary spastic paraplegia really comes into play. Because here's a disease, it's not a terribly common disease, but it's a disease that afflicts thousands of people around the world. And it's a debilitating disease that causes people to lose their ability to walk properly, to motor neuron disease. And I'm also going to talk, interestingly, about tauopathies, because I have a theory, I have a hypothesis about tauopathies, such as Alzheimer's disease, that involves these separate proteins in a very unexpected sort of way. And this is all about the issue of too little separate or too much separate. Because if you think about it, neurons contain a lot of these separate proteins. There's a lot of spastin molecules. There's a lot of ketonin molecules. And I think there's around 7 or so total separate proteins. There's ketonin-like 1, ketonin-like 2, fidgetin-like 1, fidgetin-like 2, in addition to ketonin and spastin. And the total volume of these separate proteins is enough that in the test tube would completely break the microtubules down to subunits. So it's like neurons are carried around dynamo. They're carrying around with them enough saturated protein to destroy microtubular arrays. So that demands a system of regulation. That demands controlling the cell, preventing the available saturated proteins from doing all that they otherwise could do. So with that in mind, all the important functions that saturated proteins perform, I think you can understand there's not enough saturated. The neuron's in trouble. But also, if there's too much saturated, the neuron could also be potentially even more trouble. So here's the microtubule again, just to remind you. The microtubule is this polar structure, plus and minus and. The polarity exists along the length. We'll talk this afternoon more about the molecular motor proteins that recognize that polarity and motor proteins that organize microtubules relative to that polarity. And remember this movie that I've shown a couple of times. This movie generated by Tony Brown. This is the movie showing the transport, very similar to the neurofilament transport that Dr. Roy just showed. And we talked about various features of this movement. And the feature that's most relevant to the talk this morning is that the microtubules that you see moving in these sorts of live imaging paradigms are all very short. 5 microns, 6, 7. Certainly, we've never seen a microtubule move that's been longer than 10 microns. Now, if you think about what microtubules do for neurons, particularly for axons, it's really important that they're long, that many of them are long, because microtubules form these long-distance railways for mitochondrial transports, which you've heard about. That, for vesicle transport, Dr. Brady. And also, these long microtubules are the compression-bearing struts, the architectural elements that permit axons to take on their long shape. So why is it that only we see very short microtubules moving down the axon? Well, probably long microtubules have too much drag, too much friction, and they're immobile. But the movement of microtubules is really, really important because remember we talked about during the initial formation of axons and also dendrites, it's the directionality of that transport that establishes the microtubular polarity patterns that are so important. What makes the axon an axon, what makes a dendrite a dendrite, that those microtubules are moving with their plus end leading or their minus end leading. The other crucial function of this microtubular transport is there's very little of any tubular synthesis in axons. The tubulin has to come from somewhere. Even if the majority of microtubular assembly, microtubular elongation occurs in the axon itself, that tubulin has to come from somewhere. And this ongoing supply of short microtubules supplies that tubulin. So the fate of these short microtubules is going to be either one or two things. Either they're going to disassemble at some point to yield their subunits so that their neighbors can get longer, or they're going to get longer. And once they get longer, then they become stationary until a severing protein comes along and breaks the long microtubule up into short pieces, and now once again, those short pieces can move. So that's the idea. We call the model cut and run. And what I've been interested in over the years, variety of different proteins. I've studied bundling proteins, I've studied nucleation proteins, But really, the two categories of microtubule-related proteins that interest me the most are the motor proteins that transport the microtubules, and then these cellular proteins that break them up into short pieces. So when I say cut and run, the cut is the severing event, and the run is the transport. Now, I think the severing occurs all the time throughout the neuron at some low level in order to carefully modulate the number of microtubules that are short, the number that are long, and in so doing, the proportion that are transit, the proportion that are moving. But I also think there's some key sites in the neuron where microtubular transport is particularly active and particularly important. One region is here in the cell body, particularly in the centrosome. So remember, microtubules have to be nucleated, obtain their appropriate lattice structure, and that's presumably occurring in the centrosome of the cell body. And once nucleated, those microtubules have to be released. They have to be cut, severed from the centrosome, so that can be mobilized to cytoplasm. So that's one key site. Then also, I think in growth cones, I'll show you some examples. Remember a couple days ago I showed you that stall growth cone? We had some interesting questions about that looped bundle. Well, when the growth cone stalls against that looped bundle, when that growth cone decides to start moving again, that bundle is going to break up. And little short microtubules are going to start shooting out of that looped bundle and into the peripheral regions of the growth cone. That's a microtubule severing. And then also, perhaps most importantly, is branch formation. Because the parent axon is populated by this mixture of long and short microtubules scattered throughout the parent axon. And there's just not enough of the short microtubules to somehow waylay into that newly formed branch. But rather, what has to happen is for the microtubules in the parent axon to break up, to create enough microtubules to continue to supply the parent axon, but also to supply that newly formed branch. So branching becomes a very crucial event that's dependent upon microtubule severing. Let's take a look at these severing proteins mechanistically for just a moment. What's known about these severing proteins is that they're a hexamer. So 6 of these have to get together. It's a AAA, called AAA proteins, enzymes that often form hexamers. So 6 of these have to get together, form kind of like a stop sign of an hexamer. And it's always been a little bit of a mystery, at least a couple of years ago, how it is that hexamer breaks the microtubule, because the hexamer is really a little too small to fit around the microtubule. The old idea is that maybe it would fit around the microtubule. It's an ATPase, energy-dependent event. Then it would create some torque, and it would break the microtubule. So it's too big to fit it on the outside. People thought, well, maybe it fits on the inside lumen of the microtubule. But then the question becomes, how does it get in there? So recently, fairly recently, a couple of years ago, Antonina Roll-Chat, working in Robert Bale's lab, really worked this out beautifully. And the way that it seems that it works is that this hexamer lies really on the outside of the lattice, it sits on the lattice. And what that hexamer is going to do is it's actually going to start pulling on the subunit. So in the wall of the microtubule, it starts pulling on the subunit. And then the lattice will start to give and ultimately break because you yanked out one of the tubular subunits. And that's how the microtubule severing proteins appear to work. So it's ATP dependent, and it's dependent upon this axonalization process. Now, if you, yes.
The function of these proteins means completely mechanical or they do some chemical changes also.
Well, I'm not sure. The question is this a completely chemical thing or is it a physical thing? And to be honest, I think it's a false dichotomy. I mean, I don't know that there's a difference between chemical and physical. It's a physical process of breaking the microtubule that depends upon the chemical event of ATP hydrolysis. I mean, ultimately, I think what you're going to see, what I'll propose to you anyway, is that through the process of specificity of the various cellular proteins, you're going to create global differences in the microtubule, both physical and chemical. So we'll get more to that.
Yeah.
Yes.
How many molecules? How many molecules of cannabis or plastic or whatever we need to break one molecule?
Oh, just one.
So you would put only on 6
Yeah, that's my understanding, because you really just have to tug out one subunit, just one subunit, and the microtubule will break. So here what we see is we have GFP katana, and what we see here are two different cells. One that you don't see because it's not expressing GFP katana, and then this cell is expressing GFP katana. The unexpressing cell, you see a nice, beautiful microtubule ring. Now look at this cell. Here's your GFP katana expressing cell. And the microtubules have really broken up into very short pieces. And as a result of this ongoing sagging of microtubules, notice that you really substantially reduce the total microtubule mass. So when sagging is allowed to go on, keep breaking, which can ultimately lead to a great deal of depolymerization as well. So the several proteins are very potent, very good. So we did a couple of early experiments. And what we've done here is to look at the role of katana in the release of microtubules from the centrosome. So katana was the very first microtubule cycling protein discovered. It was named after the Japanese samurai sword, katana. And way back when, this was 1998, 1999, before we were doing siRNA, before we were doing those sort of sophisticated techniques, what we did is we got a function block antibody with a lot of injection work. So you have to inject these antibodies with cultured neurons. And we used a protocol, a drug protocol. Sounds a little bit complicated, but the cell body is full of microtubules. You depolymerize the microtubules with one of these drugs. And if you rinse out the drug, you get a burst of microtubules coming right off the centrosome. And then you add a little bit of the drug back so that you don't have any further assembly. Then a little burst of microtubules, it'll jet right around the cell periphery. And that's the molecular motor protein moving the microtubules outward. So here are the images you can see. Here are the microtubules gathered around the cell periphery. But if along the way we inject a function blocking antibody to katana, the microtubules stay in a cluster. And notice in the more favorable settings, you can see the centrosome. It's there by the arrow, and the microtubules simply remain on the centrosome. So these were our earliest indications that in neurons, katana is essential for microtubules to be released from the centrosome. Now here are those stalled growth cones that we talked about. So here's the stalled growth. These are live cell images. And these are neurons injected with labeled tubule. And you can see that normally speaking, you get that moved bundle. And then when the stalled growth cone starts to move again, I hope you can see it, this is the lamellar portion. You see all those little short microtubules that they're scattered out in this lamellar region. Now look here, this is where we injected the function blocking katana antibody. In this lamellar region, I hope you can appreciate this, it's just empty. There are none of these short microtubules whatsoever. So what these two studies show in this function blocking antibody to katana is that at two different ends of the axon, in the growth cone, back in the cell body before the axon, the centrosome. Microtubule cellular autonomy is really essential for releasing the microtubules so they can become mobilized, so they can move into the axon, then out of the growth cone so that microtubules can appropriately break off of that looped bundle, become mobilized, so that the axon can start growing again. Now, my favorite idea, remember I told you the other day that I had this notion about branching of axons. My idea about branching of axons was that in order to get the new branch, there has to be a lot of microtubules separated to be able to break up the microtubular parent axon and mobilize the microtubules. So my favorite idea for a while would be if I overexpress GFP katana in these cells, I should see a lot more branch. That was my hope, and it didn't happen. So we have this axon here, we overexpress katana, and you certainly break up microtubules, your axons look a little funny, but we did not create more branches. So that was a bit disappointing.
Well, question here.
Yeah.
On the other hand, is the axon formation in length, the longest axons the same?
Is the length of the axon the same? No, if you express high levels of katana and you break up microtubules, oftentimes the axon will be a lot shorter, or you wouldn't have an axon at all.
Yes, so the question is that if you temporarily activating having an axon, long axon already formed, do you induce branching?
That would be a much better experiment to do. If you could somehow temporarily, spatially activate katana, and we haven't been able to do that yet. But let me show you a few more experiments. So what we came up with was this idea, and I'll give you the data in just a moment. But the idea, I actually presented this to you a couple days ago, is that what's happening here is that it's not just a matter of having a lot of katana available. I mean, there's already a lot of katana available, but the idea here really is regulation. And how do you regulate katana? Our hypothesis is that katana is very sensitive to the levels of tau in the microtubules. And there's a lot of tau in the microtubules, then the katana is really not so good at separating at all. So then what has to happen is that the tau has to become phosphorylated so that it's released from the microtubules, and now katana can separate the microtubules. So I would posit to you that perhaps mechanistically what's happening is that the signaling event that's giving rise to a new branch, what that signaling event is doing is it's phosphorylating tau. So that tau is released from the microtubules, and now the katana can move in and separate the microtubules. So let me show you a little bit of data that I think support that idea. These are the sorts of experiments we've heard a bit about from other studies, which we take simple fibroblasts and we use them as a workbench. So what we're going to do here is we're going to express various forms of tau. This is the 4BP, heard a little bit about this. This is tau without the microtubule binding domain. This is another tau without the microtubule binding domain here. But this is just the microtubule binding domain. This is tau without the microtubule binding domain. And tau is very, very protective. So if you express the tau with katana, you really don't see any microtubule separating at all. If you express tau without the microtubule binding domain, you see a lot of separating. So the tau has to be bound to microtubules in order to protect it against tau. So the presence of tau on the microtubule is protective. And we've actually done the same experiment with various maps. If you do it with map 1B, for example, you don't see any protection. If you have to do it with a tau family, map 2 protects, map 4 protects. These are maps with very similar structure to tau. So if we do the inverse experiment, now what we're going to do is we're going to take neurons and we're going to use siRNA to deplete MAP1B, and then we're going to overexpress ketone. Notice in the control, if you express ketone for just 12 hours, notice how the cell body reacts to the minor processes. They're much more deficient in microtubules. This is a microtubule study. Much dimmer. Whereas the axon remains pretty bright. And the reason why the axon remains pretty bright is because it's got all that tau protecting the microtubules. Notice if you deplete MAP1B, the axon's still pretty bright. Depleting the MAP1B hasn't really made any change. But if you deplete tau, now look at the axon. The axon becomes vanishingly bright from microtubules. Because now when you overexpress ketone, the microtubules have no resistance, they have no protection. This is an immunostain. This is a quantitative little scale. And what we've done here is this is a ratio that shows dephosphorylated tau to total tau. Dephosphorylated tau, remember, is going to bind the microtubules, and phosphotau is going to come off the microtubules. So notice that this region here in black, that region represents an area where the tau is particularly phosphorylated, particularly unbound to the microtubules. And that's a branch. That's a cytodependent branch formation. So another piece of evidence that supports the model that tau phosphorylation is regulating the access of ketone to microtubules. How about some other separate proteins? Let's not get so focused on ketone that we forget that there are other separate proteins as well. And this is where hereditary spastic paraplegia comes in. About 40 to 60% of all the cases of hereditary spastic paraplegia have long been known to be caused by mutations to the SPG4 gene, which encodes a protein called spastin. This is usually an adult onset disease. The average age of onset is, I think, about 42. Usually, people younger than 20, 21 are not going to be seeing typical hereditary spastic paraplegia. There's spasticity in the lower limbs, problems walking, and a degeneration is really quite specific to the cortical spinal tracts. So mutations to spastin are causing degeneration, specifically in the adult cortical spinal tracts. Very interesting about spastin. If you look at the spastin molecule, here's the AAA region. It's very similar to ketone. And that's how Elena Garley in Italy, around the year 2000 or so, took a look at the spastin molecule. And she said, that looks an awful lot like ketone. And sure enough, she made a construct, expressed it in cells, and it breaks up the microtubules. Even better. It's a very, very potent microtubule separate protein. What's curious about spastin is that there are two major isoforms. And these isoforms are produced by two star codons. So the longer isoform we call M1, that's encoded by the first star codon. And then M85 is just a little bit shorter, and that's encoded for the cells. Now, the vast majority of spasm anywhere in the body is the slightly shorter isoform. So it's the M85 or M87 isoforms. We call it M85 in mouse, rats, and then in humans it's M87. Here are the fibroblasts again. We've expressed the time for about 12 hours and spasm for equal amount of time. Here's the control. You see a lot of long microtubules. And notice there's a bit of a difference in the severing pattern. With katanin, you see a lot of the short microtubules, but you also see a lot of long microtubules. Now look at spasm. It's all short microtubules. So the properties of these severing proteins are not identical. Expressed in an identical cell type, and the resulting microtubule pattern is a bit different. Now, remember I said my dream experiment was going to overexpress katanin, and I was going to get a lot more branches. But my dream was crushed. I didn't see more branches. Well, along comes spasm to the rescue, because if I overexpress spasm, look at all these branches. This is the GFP spasm, and here's the tubulin stain. And normally speaking, here's some control axons. Well, a couple of little branches along the way. These axons are relatively short. But look at these spasm overexpressors. Lots and lots and lots of branches. If we knock down the spasm by siRNA, we severely reduce the number of branches, overexpress spasm, and severely increase the number of branches. So whereas katanin probably has a more complicated pathway to go through to create a branch, spasm appears to have a very simple pathway. There's enough spasm available, it'll get branches. If you immunostain for katanin or spasm, katanin has a relatively smooth distribution. It just appears to be equally distributed. It's everywhere. Whereas spasm, notice that you have concentrations. You have concentrations at the tips of processes, and then also here at these branch sites. Here's an impending branch site. You have concentrations of spasm. So, again, two different modes of branching. Katanin remains equally distributed. I think it's that focal dephosphorylation of tau that says to the axon, branch there, don't branch there. Whereas with spasm, it appears to be an accumulation of spasm at specific sites that yields branches at those sites. Here we see some branch sites that don't seem to have spasm. I'd say one or two things is going on at those branch sites. One is that the branch is already formed, so now the spasm is no longer required there. Or alternatively, those branch sites are katanin branch sites and didn't have to have spasm accumulation. Yes.
This spasm is what happens with several microtubules, but there is also branching of axon. So basically, only the entry of microtubule into the branch has to be decided by a lot of other factors. What is surrounding, impacting growth, or something, or is just entry of microtubule to decide that your branch is not.
The question is a very good one. Is branch formation exclusively the province of microtubule severing by these severing proteins? No, of course not. Branch formation is a complex process that requires actin-based mechanisms, membrane-based mechanisms, a coordination of cytoskeletal events. All of these work together in order to get a branch. So I don't mean to imply that simply, only, exclusively microtubule cell. And that might be another contributing reason why simply overexpressing katana, perhaps it was too big of a dream, too big of a hope, to think that simply overexpressing katana would make branches. Because it's not just the satellite microtubules, it's a combination of many, many different events. So the fact that overexpressing spastin does cause branches, I think would tell us that whatever other machinery is required to make a branch in these cultured neurons is fully available and fully prepared to do so. Is there another question?
So, how overexpression of spastin, which causes more severe microtubules, can maintain longer axons?
Say it one more time.
So, in case of overexpression of spastin, which is more severe microtubules, so how they are maintaining the length of the axon, because they are longer than the life definition are, even cadaverian overexpression.
Well, whenever you do an overexpression experiment, it all depends on the levels. The question is, if you're breaking up all of the microtubules into short pieces, how can you maintain the axon? Well, if we push this harder, we broke up all of the microtubules into short pieces, you could not maintain the axon because the long microtubules are the compression narrow struts. The long microtubules are the substrates for organelle transport. So, an overexpression experiment has to be done with a very open mind to these sorts of things. So what you need to do is you need to achieve the appropriate level of overexpressed proteins to create sufficient short microtubules in order to generate branches without destroying or altering the microtubule in such a way as to somehow curtail the other functions the microtubule array was performing.
Conceptually, a branching is being made when there is a need for it to be a means toward need. So, okay, fine. Branching is very tightly regulated by charm and pick up advantage of like sporadic. So there has to be a regulatory mechanism for spastin as well, right? Because it cannot just form branches when it's overrized, but it has to be regulated. So is it known, the regulatory mechanism for spastin?
The regulatory mechanism for spastin is really the next frontier. Maybe we'll talk about that a little bit more as we move forward toward the end, but very little is known about it. Just recently, a phosphorylation site was discovered in katana that may be important for regulating it. With spastin, that same phosphorylation site is not present. To the best of my knowledge, there is really nothing known about phosphorylation sites or other sites, or sometimes certain molecules will fold on themselves in order to auto-regulate their function. To the best of my knowledge, nothing like that is known about spastin. You have to have hexamerization. That's one level of regulation. That these individual spastin molecules were simply floating around in unhexamerized form, then obviously they do not settle microtubules. The way they become hexamerized is they use the microtubule itself as a workbench, so that they sit on the microtubule, and the presence of the microtubule enables the subunits to come together and form the hexamer. So that is one mode of regulation. We'll talk a little bit more later about the post-translational modifications of the microtubule itself, not regulating the spastin or the katana, but making the microtubule more or less sensitive to the katana or the spastin. So we have a few hints. We have the tau protection for katana. We have the acetylation, the detyrosylation status of the microtubule. But much more needs to be known about regulation and process. Not really enough is known.
So what would happen if we express all of these behaviors as so do they interact in any manner to really regulate the type of feeling?
It doesn't seem to be the case that Tom and Sebastian interact in any known ways. We used to wonder a little bit, given that they're similar, could they say co-hexamerize? I don't think they co-hexamerize because their distributions are so different. And we used to wonder, for example, with an only function blocking antibody, because the phenotype was so strong, might that antibody also inhibit spasm in addition to the phenotype? At present, again, to the best of my knowledge, nothing is known about common spasm regulating one another, except maybe one thing. That if you knock out one of them, say you make a genetic animal that knocks one of them out, and you use one of the siRNA procedures to knock out common spasm, I think the levels of the others will come up to compensate.
So I have two questions. So, is there any difference in cellular localization of the two spasm? Because when you show the isoforms of the spasm, they have NLS. One isoform has NLS, NES, and NLS, and another one has only one NLS.
I will talk about the two isoforms in a bit. You cannot discern the two isoforms with separate antibodies as of yet. We're trying to develop separate antibodies, but it's a tough challenge. I think the properties of the two isoforms may lead to different distributions. One of the isoforms, the full-length isoform, for example, has a very highly hydrophobic domain, such that you might think, going back to Dr. Roy's talk, you might think that one isoform, the M1 isoform, is conveying fast axonal transport, whereas the other isoform is transported slow component B of axonal transport, which might lead to very different distributions. But also keep in mind that the longer isoform is really only expressed at any detectable levels under very certain circumstances. And I'm going to talk about that very shortly. But the vast majority of this spasm is just a shorter isoform.
So, another thing is that in case of overexpression of spasm, can we speculate that because the microtubules are now short, there is redistribution of proteins to regions as a result, microtubule and the protein that it is?
It's a very good point. If you think about the manifestations of the microtubules being short, and maybe a redistribution of proteins. Now, tell me the redistribution of maybe one of the most important proteins. You're going to hear a lot more about the so-called plus tip proteins tomorrow. But you've got a lot more plus ends. That means you've got a lot more plus tips potentially. And the plus tips really have the property to interact with cortical structures. If you have EB1, EB3, they can interact with other plus tips, the clips, the clasps, they can get captured. Remember, one of the two presentations was all about the role of the plus tips in the growth. So I suspect that maybe what's happening in branch formation is it's not just the mobilization of the short microtubules. It's the fact that you have so many more plus ends and the accumulation of all these plus tips, and then those sorts of things are essential. So these are wonderful questions. Remember, nothing in biology happens in isolation. You don't just knock out a protein, or overexpress a protein, or break up microtubules. Only one thing happens. Oh, now they're short, now they can move more. Sure, a lot of other things are happening as well. It's a very good
Does branching trigger severing triggers branching?
Does branching trigger severing, or does severing trigger branching? I would say severing triggers branching, for sure. But I wouldn't dismiss the possibility that branching could induce severing as well. I use, say, for example, you start to form a branch and get that actin-based structure pulling and pulling. It used to be a microtubular literature called bending and breaking, that what people would study was David Moley or Claire Warren Stores, and these folks, microtubular people, would look at the leading edge cells and get the impression that because the actin structures are pulling on the microtubules, they get a phenomenon called bending and breaking. Well, in neurons, microtubules bend all the time and they don't break. I don't think microtubules break because they bend, but it may be that by virtue of bending, you enhance the access to the cellular proteins. So I think it could potentially go both ways, so I like that question. Now, what really causes branches to happen? What causes branches to occur are these complex pathways. And there are growth factors that will induce branching to form. And one of them is called basic FGF. FGF is fibroblast growth factor. And it's a very potent factor which will cause a lot of branches to form. And I know that your eyes may not be able to appreciate all the details here, but this is a lot of Western blotting that we did. And to make a long story short, if you take cultured hippocampal neurons and you treat them with FGF, you get a lot of branching. Mechanistically speaking, what's happening? Well, you get an elevation in catanin levels, you get an elevation in spastin levels, and you get lower topics formation. So really, at 3 different levels, we were wondering, well, maybe FGF causes branching through a spastin mechanism, or maybe FGF causes branches through a catanin mechanism. Maybe it's through the tau pathway. And as it turns out, this particular growth factor really causes changes consistent with branching in all 3 of these. There are a lot of factors that cause branching. There may be, natrins or different things. And I think if you survey these different factors, you may find that certain factors work through a spastin mode, and other factors work through a catanin mode. But at least at present, it appears to be that this factor works through both. So here's the slide I showed briefly a couple of days ago for our 2 modes of branching. Spastin mode, which appears to be an accumulation of spastin at particular sites that give rise to branches, and the catanin mode, it works through that tau phosphorylation that we discussed. Yes, that's your question. No. Okay. So what else besides tau might regulate these cellular proteins? And one of this is this tubulin code that we brought up the other day. So maybe certain of these cellular proteins have a preference for acetylated microtubules or deuterosylated microtubules. Nature tends to retool, repurpose, change things around a little bit, but utilize those themes that are so very effective. That's why we see time and time again in nature very disparate types of cells, very disparate organisms on the evolutionary tree, really tapping into very similar mechanisms, very similar approaches that may on the surface seem like they're not all that terribly similar, but in actual fact, they really are. And if you look at the mitotic spindle, if you look at the microtubular neuron, on the surface, they really seem quite different. So you see behind me, you've got a mitotic spindle, and then you've got neurons. Spindles have these duplicated centrosomes. They have microtubules that are leaking out. They've got this overlapping region. These are astral microtubules. They have uniform orientation. These microtubules in the midzone have non-uniform orientation. But if you look at neurons, they don't look anything like mitotic spindle. At least not on first glance. If you look more carefully, I think you'll see some similarities. For example, these astral microtubules, with their uniform orientation, they're a bit like the axon. These midzonal microtubules, with their mixed orientation, they're a bit like the dendrite. So we initially saw some proof of principle information, some proof of principle that maybe the underlying mechanisms of the neuron are really quite similar or borrow themes from mitosis. And I'll tell you, the more we've looked into this over the years, particularly with regard to molecular motor proteins, we found that not only are themes borrowed, really, it's the same motors. That these motor proteins, I mentioned, I think, way back in the first day, that I heard about from my graduate student, that there are motor proteins that are specialized to transport microtubules against microtubules, microtubules against active filaments. But those mitotic motor proteins, well, they exist during cell division, but that's all. Every time we look for a mitotic motor protein in terminally post-mitotic neurons, we find it's there. Every time we inhibit it, we've got a major phenotype. So the very idea that these molecular motor proteins are mitosis specific is false. They're not mitosis specific. They've been really retooled in a different way to give rise to the molecular limited microtubular base of the post-mitotic neuron. The very first motor that we need, let's think about the microtubular base of the axon. The very first motor that we need is a motor protein that would transport microtubules to the placenta. That's what you need for the axon, and that's what you need to get the dendrites started. Now, cytoplasmic dying is really perhaps the most famous molecular motor protein. It's used for many different purposes. Kinesins are more specialized for doing different things. Dying is a very multifunctional motor. And dying is most well known as the motor protein that moves toward minus ends of microtubules. So it's known as the retrograde motor that conveys cargoes retrogradely in the axon toward minus ends. Remember we talked about all motion being relative. Here's your dying molecule. Here's your little cargo. Let's say we have a very long microtubule up here. Then that dying is going to go toward the minus end of the microtubule and carry that cargo with it toward the minus end. But let's say that this cargo is not a little vessel. Let's say this cargo is a long microtubule. Let's say the cargo is the actin site matrix. Then what's going to happen with a smaller microtubule? The microtubule, all motion being relative, and the microtubule will move with the placenta leading. Because that motor protein, that's struggling to get to the minus end of the microtubule. It's trying to move toward the minus end of the microtubule, but it can't because the cargo won't move. So it's the short microtubule that will move with the placenta leading. So dying is a very attractive candidate for us for the motor that moves microtubules, the placenta leading down the axon. There's also precedent. Another place you find dying very prominently is with those astral microtubules in the spindle, pulling on those astral microtubules against the actin of the cortex in order to divide the two path spindles. So there's precedent in the mitotic spindle for dying generating these kinds of forces. So what we did is we used our siRNA approach. So this is small interfering RNA using cultured rat synthetic neurons. And we depleted cytoplasmic dyne. Cytoplasmic dyne has 3 different supplements that are each one of which is a different primary gene product. So as long as we deplete any one of the 3, then we disable cytoplasmic dyne. So we depleted the heavy chain, and we depleted it using siRNA by about 90% or so. A couple of reasons. One is that just technically speaking, sometimes you can't deplete it all, but also we depleted it more, but we didn't really have an axon we could study. It was a little too abnormal. But what we found here, this is a couple of different days, and remember that normally speaking, about 2/3 of these short mitochondrials move anterogradially, and normally speaking, about 1/3 move retrogradally. Now here, look at the dyne depleted at 4 days and 7 days. It's about half the number. And then in the retrograde direction, it's not been affected at all. So we took this as evidence that our hypothesis is essentially correct. That cytoplasmic dyne is an important motor protein for transporting microtubules with their plus ends anterogradially moving down the axon. Now, why is it only half? Well, maybe it's because we didn't deplete the dyne completely. Maybe there's another one protein that shares that responsibility with dyne. That's not unusual in nature. That nature would invest everything into one protein. Sometimes there's a share of proteins. But also, I think sometimes, particularly with motor proteins, there's some unusual things that go on. For example, if one motor protein is accustomed to moving a particular cargo, and you take away that one protein, it may very well be that a different motor protein that normally does not move that cargo would suddenly start moving it. So I think when you interpret these sorts of data, you've got to interpret them with caution. So I think we have enough data here to have some confidence that dyne is among the motors that transport microtubules anterogradially down the axon. Might be the only motor that does so. We still need more information. Now, one of the themes that I indicated in one of the earlier lectures is I think these motor proteins are pretty smart. And to those of you who study motor proteins, I don't think you'll be offended by this. I don't think they're smart enough to say, here's a short microtubule I'll give you, but long microtubule, no, I'm not going to bother. I think they would attempt to move long microtubules, but they can't, because a long microtubule has too much friction, too much drag, for whatever reason. But those forces are still highly active. They're pulling and pulling, and they're trying to move those small microtubules. We think that there's a variety of forces. It's like an isometric exercise, pulling and tugging both directions on those microtubules. So I think that the short microtubules serve the functions we discussed earlier today in terms of orienting microtubules, in terms of delivering subunits, in terms of acting as nucleation sites. But they're also a harbinger. The movement of the short microtubules is an indicator, a bellwether, a harbinger of the forces that are on every single microtubule. So think about the microtubule, long, short, every length, is being under a whole series of forces. And those forces, I think, are what are responsible for the structural integrity of the axon. So that you think of a building like this, what's responsible for the structural integrity? It's the nails and the mortar that hold things together in a permanent way. But living cells aren't hammered together in a permanent way. They're more like machines. They have sliding parts in them. So I would argue it's not so much that the cytoskeleton is mortared together, but these forces are what are integrating the microtubule into a functional unit. So the same motor proteins, as I march through and talk quickly, will be relevant to the short microtubules as well as the long microtubules. And I think these forces are manifest by these experiments. If we deplete cytoplasmic dynein, the axons become much, much more retractable. So we're here, we're exposing to semaphorin or nitric oxide or something that makes axons retract. When dynein is depleted, look how much more retractable. Normally a little bit of retraction. Now, not only are they very retractable, look at the microtubules inside, look how contorted they are. Some people, before we did these studies, would suggest that microtubules would just disassemble. They just go away when axons retract. But no, look how they contort. Those microtubules are under a lot of forces. That's the message here. So if you take away one of the one proteins, dynein, that in addition to transporting short microtubules, it's pulling on the long ones and keeping the axon out. Look how retractable it is if you take away those forces. Now, you might wonder, what are these forces being generated against? And I think it could be the long microtubules. I think it could be the active side of matrix. We have some data to suggest that it could be both. The whole idea here, and I think I alluded to this in an earlier lecture, is that this is your active side of matrix. It's highly compressible. It's got myosin 2 associated with it. So left to its own devices, the axon will retract because it's got these very powerful actin-myosin forces. The dynein, by pulling on the microtubules and attaching to, in some places, the actin cortex, is opposing that attraction. So that the dynein forces win, the axon grows. If the myosin 2 forces win, the axon retracts. So this is an ongoing competition of these forces. And this could be regulated by things like phosphorylation of myosin 2, complementary chains, things like that. Even regulation of dynein from phosphorylation. Things that would control which of these two very strong forces. Growth cone turning, I think, is a variation of this theme. Because you've got this actin and red that's undergoing a constant retrograde flow, constantly pushing back the microtubules. And I think what's going on here is the dynein forces are serving to drive microtubules out. So they're helping the microtubules to swim upstream to fight back against the actin myosin. So again, opposing forces. And which side wins the argument, I think, is what's going to be crucial for if the growth cone is able to turn, because those microtubules have made the fast side of the growth cone.
Would the situation be different in case of a developing neuron and a developed neuron, as in the growing neuron versus one which is to be just maintained?
Wouldn't there be a difference between a growing neuron and a what?
And a neuron which has already attained its length and...
Yeah, it's already attained its length. Yeah. We're talking here primarily about development at this point. The issue of when an axon reaches its target, and particularly in the case of regeneration, you've got a different scenario. You've got less filamentous actin, for example. Yeah, so I think not enough has been done in the adult nervous system. I think much of what people are finding out in regeneration feels a little surprising. Things aren't always the same as development. So yeah, I think it's a good.
Yeah. What about the evolution of the normal? Are they all related safely, or they are like
are all of the long microtubules of the axon oriented in the same way? Is that what you're asking?
Yeah.
Yeah, the axon is known for having a nearly uniform microtubule polarity orientation. And that's really the theme of my talk today, is how does the axon achieve its uniform polarity orientation, and how does the dendrite achieve its mixed orientation? So that's really the entire point. I'm not going to tell you that every last microtubule, every single one has that uniform orientation, because we have seen an occasional microtubule that's flipped in its orientation. So my interpretation of that is that the axon is constantly at risk of corruption, that there is a possibility of microtubules flipping, so that there has to be a constant clearing mechanism. So my theory of direction radially moving microtubules is the reason why a population of microtubules exists is those are maloriented microtubules that are being cleared as part of the preservation mechanism to ensure the microtubule pattern stays almost uniform. So getting back to growth hormone turning, if we challenge growth hormones to turn, this is a polylysine substrate, this is laminin. Laminin is much preferred, so the growth hormone will turn to stay on the laminin. This is an immunostate, microtubules in purple, active in green. And notice how the purple microtubules are invading these photopodia. This growth hormone is going to turn. This is our dining heavy chain depleted. And notice the microtubules remain knotted up in the growth hormone. They do not invade the photopodia. The growth hormone just stops. Now let me show you the live cell imaging. These are the EB3 columns that enable us to visualize the placenta. This is a control. I showed you this a few days ago. See a lot of microtubules invading growth hormone. Now here's the dining heavy chain depleted. Now here's the, I don't know if you can see, here's the periphery, way up here. And notice those microtubules are just struggling to get up. They're just struggling because now they don't have the dining forces to really drive them up. They just have myosin 2 forces that are pushing backward. They don't have the myosin 2 forces. So I think these are a couple of examples of this antagonistic force relationship between dining and myosin 2. Axon retraction and growth hormone turning. A tug of war. But what about other hormone proteins? I think this antagonistic relationship between myosin 2 and dining, that's the fundamental. But what if you really want to make growth hormones turn? What if you want to modulate axon retraction? What if you want to have an extra degree of control over short microtubule transport? That's where I think these mitotic hormone proteins come in. And the first one we looked at is kinesin 5. Kinesin 5 is a homotetra. It's got 4 motor domains projected upward. And it's really important for mitosis. In fact, if you inhibit kinesin 5, you get a monastral spindle. It just collapses. And that's why the prototype drug is called monastral, because it collapses the spindle. The drug industry, pharmaceutical industry, really put a lot of effort into making anti-kinesin 5 drugs because it just seems like an ideal choice for cancer therapy to stop cell division. And if you look at some of the web pages that the pharmaceutical industry has made over the years, they really brag that, oh, there could be no ill effects in the nervous system because there's no kinesin 5 in neurons. Oh, yes, there is. Oh, yes, there is. So I'm going to show you some dramatic phenotypes that we get when we treat neurons with monastral. Or here's a Western blot to show that with siRNA, we're able to really clear out almost all the kinesin 5 quite effectively. Very interesting phenotype, surprising. If we inhibit kinesin 5, control in yellow, experimental in red, the rate of axon outgrowth is much, much faster. The axons grow faster, the growth cones are big and nice, the axons are beautiful, they're incredibly mapping, they look better than controls. and they're more branched. It's kind of rare that you would not know protein axon or happy. Now, when we looked at microtubule transport, I have made the guess that maybe PC5 is the retrograde moment that moves the microtubules backward. So maybe the reason why the axons are going faster is because you only have a hereditary transport. No, that wasn't the case at all. Again, control in yellow, experimental in red, and if we either treat with monasterol or the siRNA, the frequency of short microtubule transport doubles in both directions. You get more microtubules moving forward, more microtubules moving backward. And this is only microtubules. It's not mitochondria, it's not vesicles, it's only microtubules. I think what's going on is that kinesin 5 is breaking. It's a very slow motor. And there are certain things that can make it even slower. And this is known for the mitosis. So I think it's a break. It's a break on microtubular transport, which manifests as a break on axonal growth. You take off the brake, a lot more microtubules can move, and the axon grows a lot faster. When axons grow, you can see here, this is control, you get a lot of these little tiny branchlets forming, but most of them retract. But notice here with the monasterol or the siRNA, you get these little tiny branches forming, but they don't retract. They don't retract. So kinesin 5 inhibition is stopping retraction occurring. That's why the axon looks so much more branched. Here's that turning piece. The growth cones grow on the lamina, they hit the border, and they turn. Here's monasterol. They go right across the border. They go right across the border. They do not turn. They don't stop like dying inhibition. They cross. So we're seeing differences in microtubular transport, in retraction, and growth cone turning. The same family of phenotypes that we saw with dying inhibition. Different results. Here's another example of the turning. We have here an NGF, growth factor coded B, the growth hormone normally turns toward it. Here, NGF coded B, and the growth cone in the presence of the monasterol drug, it just ignores it. Here you can see the movement control. And now with monasterol, it just ignores it. I think this is all regulated by phosphorylation. Kinesin 5 has a phosphorylation site. And with a phosphospecific antibody, you're able to see it's better polarized. See, it's kind of on one side of the growth cone. You see the microtubules bursting through one side where the phosphokinesin 5 is now localized. I don't want to take a lot of time on this because time is short. But here you can see a growth cone approaching the border. You don't really see any phosphokinesin 5. Notice it kind of pops up here when the growth cone hits the border, and then boom, it turns the opposite direction. So kinesin 5 doesn't break. It's pushing back on the microtubules on one side of the growth cone so that they can invade the other side of the growth cone, and then the growth cone can turn. So I think that this is tipping the balance. You constantly have that myosin 2 diamine competition kind of pushing at one another. But rather than having the growth cone turn by altering diamine forces, what you really do is you add this extra factor. You add kinesin 5 to the mix. And that's the decider. That's the decision maker. The kinesin 5. Yeah.
This thing is irregular in shape. The growth cone is very irregular in shape, so the force balance must be satisfied very locally everywhere. Does that make sense?
The growth cone is irregularly shaped, so the force balance.
Must be satisfied very locally.
Yeah, that makes sense. So this is kind of our model, the way that we think about this, is that you have dying forces kind of pushing out, myosin 2 forces pushing in. Adhesion 5 is here in the transition zone of the growth cone, and it's phosphorylated on one side to act as a brake so the microtubules can only push through on the other side and the growth cone turns. Now, as a test of that, we use microtubules. And really, to make a long story short, what you do with microtubules, at least this variation of microtubules, is you express GFP kinesin 5, and that's going to incorporate into the homotetramer, so that some of the homotetramers will have kinesin 5 GFP. Then we're going to use a strong enough laser in order to ablate kinesin 5 focally in the growth cone and see what sort of results we get. So here we've done that. Here's the region. You can see those EB3 cones. So this is an experiment done by Vidya Gadar, who was an Indian student in my lab. It's superb work. This I can only imagine how much skill it took her to do this, where she's expressing the EB3 cones, so she can see them here. And then, if she does the laser, and she ablates kinesin 5 on this transition zone, notice now you get a lot more cones breaking through. So here we do a reverse image. This is before the laser. Hardly there's one comment here, then after the laser, a lot more through. And she only got this result in the transition zone. If she conducted the laser, she focused the laser on the central domain, which is here, or the peripheral domain here. This is not the case. Only in the transition zone where kinesin 5 resides. So this makes sense as a brake, because it can really rapidly restrict the entry of microtubules needed for growth cone turn. Now that you put the myostrol in micropipette, notice that the growth cone will turn toward that pipette, will turn toward the myostrol, to broken down break in that region. So you can think of this as a tug of war, where the kinesin 5 joins the myosin 2 side of the equation against the dying. That's one way of looking at it. But maybe a better analogy is to think of kinesin 5 as a brake. Because if you don't have a brake, your car can drive really fast. The axon can grow really fast. But when it's supposed to turn, guess what? It doesn't turn. And then you're in trouble. So I think that's a good analogy. Now what about other mitotic kinesis? That's 5. There's another one called 12. We looked at 12, and some elements of the phenotype are really quite similar. If you inhibit kinesin 12, you have to use siRNA. You get longer axons, but they're not more branched, they're less branched. You get a failure of growth cone turning. You do get a substantial increase in microtubule transport. Many things are like kinesin 5 inhibition, some things are different. Here's your increase in microtubule transport, both directions. That's really even greater than 5. And I think the reason why certain things are different, certain things are the same, is that kinesin 12 has a myosin-like domain. So we do these pull downs. Kinesin 12 will pull down an actin. Kinesin 5 will not. So things like branching, things like growth cone size, growth cones get bigger if you inhibit kinesin 5, they get smaller if you inhibit kinesin 12. I think that's because of the actin domain specific to kinesin 12. Now, what about kinesin 6? Kinesin 6 is a very interesting one. Again, a mitotic kinesin. It's localized here in the midzone. In neurons, it's in cell bodies, and it's in dendrites. It's not in axons. We can't pick it up in axons at all. But here's a very curious result. If we inhibit kinesis 6, we deplete it with siRNA, control axon, and look at these guys, very, very long axons. So the axons are getting longer. There's a higher frequency of microtubule transport. And yet, it's not even in the axon. So why does depleting a motor protein that's not even in the axon, why does that make the axon grow longer? And why does that increase the number of microtubules in transit? I think it's because here's your cell body. And I think at the level of the cell body, these molecular motor proteins act as a traffic cop. And the way I think about this is that axons and dendrites are not conducting their business separately, but they're conducting their business coordinately, with the cell body being the control cell, the traffic cop. So you've got, at the level of the cell body, these microtubules that are using dynamo, they're being transported into the axon, using dynamo, but they're not going as much as they otherwise would because they have these 5, 6, and 12 holding them back to some extent within the cell body. And if you got rid of 5, 6, or 12, then more of them get into the axon, and the axon will grow faster. Now, certain elements of their functions, like growth co-turning, for example, that's local. But the flow of microtubules, the number of microtubules in transit, and correspondingly the length of the axon, I think I control, at least in large part, not entirely, but in large part, at the level of the cell body. And that really brings dendrites into the story, because what I'm going to propose to you is that maybe these motors are not only brakes, but they can accelerate these microtubules in the opposite direction. So here's an older neuron, sympathetic neuron culture, and now you've got nice, thick, tapering dendrites, and the axon has gotten so long, it just fills the culture niche. If we deplete any of these mitotic motor proteins, either before the dendrites have developed or after they've developed, look what you get. Those dendrites have thinned way down. The minus distal microtubules have moved right back into the cell body. And those dendrites have become, for all practical purposes, they become axons. Morphologically, structurally, they become axons. So these mitotic motor proteins are really essential for driving the minus distal microtubules into the dendrites and keeping them there. And we did this with the current technique. So again, here's our movie. Showed you this a few minutes ago. These are all plus microtubules. So here's your axon. These are nascent dendrites. And plus distal microtubules. This is probably primarily diamine forces. And you've got those mitotic motor proteins in the cell body that are holding back some of the transport. Now imagine this. Those motor forces are getting stronger and stronger and stronger until now they get so strong they literally back these microtubules up, minus them leading into those other processes. So they now require mixed orientation. And that's what I think is going on. So something that doesn't look very much like mitosis on the surface is really utilizing mitotic themes, mitotic motor proteins, in order to set up uniform and non-uniform polarity patterns in axons and dendrites. And this just shows an example of elucidate. One of these motors is kinesin 12, really building up the dendrites as they develop.
So there must be a set of stable microtubules near that area which is not going to the area against which these microtubules are related.
Yeah, so for any microtubule to move, it has to move against something. And that's why I think it's really important in these developing dendrites that first you have the plus end distal microtubules, so that the minus end distal microtubules are using the plus end distal microtubules as their substrate against which they push.
Question.
Yes.
So in that case, the original axons become shorter.
The original axon.
Where the dendrites, so in the siRNA context, and the dendrites also become an axon. So the original axon.
The one that is initially extended as an axon.
No, I don't think it becomes shorter. Why do you think it might become shorter?
Because then the opposing forces are. It's not there in the original form.
Oh, well, you know, at that point, the axon is so incredibly long in the ditch, it'd be probably kind of hard to find out, but could be. It could be.
So how is kinesin 5 itself regulated? What brings it, you said kinesin 5 is found in only certain areas. How is it regulated?
Okay, that's a great question. How do these molecular motor proteins find their appropriate place? Kinesin 6 is only in dendrites, not in the axon. Kinesin 5, and then also kinesin 12, notice how it builds up in the dendrites. So it's not as dendrite specific as 6, but it gradually builds up. This is reverse image black, it indicates a higher content. And you know what? 5 does the same thing. Kinesin 5, that's so important for axons. Once you get dendrites, it really starts accumulating in dendrites. And I think there's a couple of reasons for it. One is most of these motors are regulated by phosphorylation. Kinesin 5 certainly is regulated by phosphorylation. Phosphorylation site that determines whether it associates with microtubules. So just like tau, just like MAP2, the phosphorylation that occurs in dendrites and axons is not the same. Certain proteins are phosphorylated in one versus the other. The other reason is what we've been alluding to about the tyrosine state, the acetylation state of the microtubules. Kinesin 5 much prefers tyrosination, tyrosinated microtubules. And there are two places in the neuron where microtubules are most tyrosinated. One is dendrites, and the other is growth cones. That's where it is.
You mean details?
Tyrosinated. Tyrosinated is the unmodified form, and that predominates in dendrites and growth cones. So this is what I showed you with the movies. We call it a co-regulation. It's at the level of the cell body. You have all the forces of these three mitotic motor proteins that are collectively suppressing the transport of microtubules in the axon, not completely, but attenuating the transport, so that when those forces build up, they can really drive the microtubules to dendrites with the opposite orientation. But two of these three, 5 and 12, are certainly actively axon as well, contributing to axon growth, to Baas forces, branching, and then also growth control. Now, is this theme maybe even a little bigger than axons and dendrites? Now, I want to just very briefly bring up neuronal migration, because I have another Indian graduate student, she just got her PhD at Bombay, and she's been studying neuronal migration. And here's what she sees with kinesin 5 inhibition. This is a migrating neuron control. Now look at this one. migrating with a piece of 5 outside RNA, it migrates much faster. Much faster. So we hit a piece of 5, and just as axons grow faster, neurons migrate faster. And then this is a piece of 6 inhibition. This is a control, migrates forward. Now look at this one, piece of 5. It's not just slower. Look at this guy jumping around. Migratory neurons are supposed to be bipolar, the trailing process and the leading process. It keeps poking out every which way, and it doesn't know where to go. It's totally confused. So, this is kinesin 6. Kinesin 6 is crucial for cell division. It orchestrates cytokinesis. And here we see it absolutely essential for migratory neurons to behave properly, to become bipolar. They're quintessential morphology, they're directed migration. So, again, people would never even have dreamed of this not too long ago. That mitotic motors, they have no business, in theory, being internally post-mitotic neurons. They're so essential for what they do. And I think it just goes to illustrate that balance of forces on the microtubules is really so essential. And she's also done taking us to in vivo, doing the in neurodegeneration, just really done beautiful work. So the big message here, conservation of feeding from the mitotic spindle to terminally post-mitotic neurons. These are the folks who did the work and collaborated with me. And this is Vidya and TV. So I think India is a wonderful gift that is given to me. So thank you very much for your kind attention.
I think we have a lot of time for questions.
Are you all questioned, huh? Oh my gosh, I've had so many questions. Any other questions? Are you eager for your excursion? Well, let me just take a moment, say a couple of things. I have never had such a wonderful audience as this group. Never. Students here, you're thinkers, you think before you speak, and your minds are working, and your eyes are attentive, and it's absolutely wonderful. I've had such a great, terrific time. And I'm so in admiration. I wish the students in the United States, we've got some really good students in the United States, but the passion, the scholarly nature with which you take your work and the interest that you've shown, I think is so incredible. And I think it's also a tribute to your wonderful professors, that you're as educated, passionate, and also I think as collegial and good-hearted as you are, because I think that's also really an important part of science, because science can't exist when people are alone in islands. It just doesn't work. You've got to be able to make friends and interact with people and project a positive attitude, forge collaborations, attract students to your lab. So I think really the good heart, the goodwill I see on display here is such a tribute to all of you and your wonderful professors. So I want to thank you very kindly for
Okay, since we are doing pretty well, we are actually back on schedule now. So Ambarish will be the next